woken up to the benefit. Now, you showed that paper, LDN and cancer.
Yep.
When I started writing these scripts out for cancer patients, I was told I was the only one who was writing low dose naltrexone script for cancer patients. And after that paper was published, he now tells me that there's many doctors writing it out. So they know, and they know there's a benefit. So there should be a forum for approving this.
And there should be a way of collecting this data prospectively. It's basically a stage 4 clinical trial, isn't it?
It is, and
which is better than a stage 3 clinical trial.
It is much better, and this is why it's interesting mentioning lenalidomide. Lenalidomide had stage 4 approval in France before it was ever approved here, using the stage 4 process, because it was seen to be such an unmet need, and of course that stage 4 process proved it.
But the regulatory agencies don't take that kind of data. They look for the stage 3 data.
Yeah,
the $20 million RCT industry funded data.
Yeah, well, we now know the only reason for doing that is to keep anything useful out of the patient's hands. I mean, I hate to be so cynical, but my experience leads me to
it's completely realistic.
Yeah. And so the other thing is that, a stage 4 study is much better than a big randomized study for these agents which are freely available for everybody.
Yes.
Why do you need to? I've something else which I'm not going to mention on this. I'm not going to complicate any further. But something I thought might help for the joints. It was recommended by a patient, so I tried it, and so I tried it. It improved. I stopped. The problem came back. I took it again, disappeared. I did this randomized, non-controlled trial on myself and came to the only conclusion was this was really superb for me. I didn't need to wait for a randomized controlled trial of this thing, which is never going to be done, by the way.
Because all the basic toxicology, the stage 1 and stage 2 stuff's already been done.
Yes, all that's been done. Yeah. And actually, we've also done that. Why is it 4.5 milligram and below, and not 4.5 milligram and above? Because they're two different drugs. The way they're absorbed, all drugs come in two forms.
It's the same molecule.
It's the same molecule, but in a different form. And the first form is absorbed up to 4.5. After that, it's drowned out by the other molecule. You have left molecules and right molecules. This became very, very important with thalidomide.
Because I see. It's the chirality.
Chirality. They found that one of them was the cause of the birth defects.
Yes.
So they very cleverly spent, and I spoke to the guy who actually did all this work. And they then converted it and made it just in the right form.
Yeah, all L form or D form, whatever.
Yeah, exactly. But as soon as they gave it to various animals and things, but as soon as they gave it to humans, the human liver immediately converts it back to the left.
So the left was the teratogenic form, was it?
Yeah, I can't be 100% sure.
Yeah, yeah.
I don't want to go down as...
Yeah, sorry, it's a Tuesday.
I would have to check with it because...
But anyway, you can give right thalidomide and the human liver will convert it into the teratogenic form of the
exactly. And that's what this guy sat down and told me. He said, this is why, and I used his evidence to put pressure on them to make the analogues, because I said it's pointless going down the chiral path. This guy's already done it. It's a waste
Just out of interest, the lenalidomide, is that teratogenic?
No, it's not, but it has to be treated as such because of its close relationship with thalidomide.
Yeah.
In fact, it has very few side effects compared with thalidomide. When I was doing a lot of work with thalidomide, I was the one I told them, I told Celgene, I said that we have a big issue, a big problem, and this is how I got the analogues done. I said, I'm not worried about heterogeneity, but it's very clear for conditions, chronic conditions, cancer, you need to give this drug for a long time daily. But everybody starts getting neuropathy, whether it be 6-8 weeks. It's absolutely, you cannot continue with it. So you must...
This is with the lenalidomide.
Yeah, yeah. No, so that's what sparked them to finally make all these drugs. And again, we just screened them all, and lenalidomide just ticked all the boxes.
Oh, so you don't get the neuropathy with the lenalidomide?
No,
You do.
and then when it went into the clinic, this is the first thing I was looking for.
You don't get it.
You don't get it, and actually, people who had neuropathy from thalidomide said it actually improved when they went to lenalidomide.
So the thalidomide causes neuropathy. The lenalidomide does not cause neuropathy.
No, it does not. No.
But it does treat myeloma and lymphoma.
Yeah, it's even better than thalidomide in inhibiting all the main pathways. And this is another really important thing. Lenalidomide, pomalidomide, they inhibit so many pathways. They inhibit inflammatory, angiogenesis, there's a whole range. And so much so that Celgene told me to stop looking because they only wanted one good explanation.
Yeah, but it fulfills Bradford Hill really nicely, doesn't it?
Yeah, but what is interesting from that book, that paper that you showed that we did, low dose naltrexone is exactly the same. The more we look, the more pathways it interferes with.
Well, I've got 4: arresting tumour growth, increasing apoptosis, increasing immunity, and reducing inflammation.
Mm-hmm. I mean,
and another one, it seems to be that it promotes gene expression of apoptotic.
Yes, this is the
proteins.
That's right. This is the brilliant work that Wei Lu did.
Amazing.
He did all this off his own bat.
He sounds like a really useful guy. This.
Oh, he's fantastic. He came to me having had a big history and past in looking at scientific aspects of the cannabinoids.
Yes, I've read some of those papers.
Yeah, it's really, really good. And he was able to show that in some cases there's synergy between LDN and the cannabinoids. And more importantly, due to what I thought there had been a big error in the lab, he showed that the cannabinoids have significant anti-cancer activity only if they're used intermittently. So
isn't that interesting?
If you take them daily, nothing happens.
Is that true? Is that true for the LDN as well, for low dose naltrexone?
Well, people have posited this, that this might be better 3 days on or 3 days off.
Really?
My argument is I've always used it daily at the low doses, and at the low doses, it's only in the blood for a few hours, so it is intermittent. It is past already.
Yeah.
That's my argument. But with the CBD stuff, and we did it in a range of cell lines, it's very clear. In fact, I thought they'd got everything mixed up when they gave me all the results, because the cell lines that were dying faster and quicker were the ones that had CBD for 3 days and then withdrawn. That was just meant to be using as a control for the effect of washing out the nutrients, the food for the cells. They do it. It's a regular thing to do. And I said, this is unbelievable.
So, sorry, say that last bit again, Gus. I didn't. Why is it?
When you're looking at cell death, say over a month, you have to sort of keep the food and liquid and nutrients up. So you sort of wash the cells out and put them in fresh nutrients all the time.
Yes.
And so when you're looking at the long-term effect, you give them their food and water basically, and you continue the CBD. But just to see whether that process has any effect on it, you wash it out and see, or to see whether the benefits short-term or long-term. But it was so clear, 3 days followed by washout, then no more, the cells go into apoptosis like you wouldn't believe. If you keep the same dose of CBD up, the cells just go a bit slow. They don't die.
Have you got?
They slow down.
It's a bit geeky, but is there a mechanism for that?
Well, my only explanation, and if other people have got one, I'd love to hear it and be explained. But my only gut feeling explanation is we're watching cold turkey here. The tumour cell lines have got rapidly addicted to the cannabinoid pathways. And in cells, the opiate and the cannabinoid pathways, they're really big, important pathway cells. So they've got very highly addicted to it, and then you stop it, and then they go into apoptosis. I mean,
amazing. So there's something about the intracellular biochemistry which becomes tolerant to the presence of a particular molecule.
Yeah. So once again, it opens up, and that's another reason. Like there's several other drugs which might be better off in low doses and intermittency. I think it's a whole new potential of the pharmacology is. As I said,
it really is a whole new way of thinking. That to me,
it is, yeah. And to me, it's just incredible, having been brought up, the more is the better, because that is oncology. In fact, we don't, when you develop a drug, basically, chemotherapy drugs, there's a thing called LD50. I mean, they keep up
lethal dose,
50% of the mice are dead, and then they start around that and reduce it which is why so many of them are so toxic, is because they're developed like that. And I would say that there's no need for it.
No, I agree completely. But we have the same with aspirin as well. Post-MI, for example, post-heart attack, 300 milligrams of sublingual aspirin is more effective than 600 milligrams of sublingual aspirin. So
absolutely,
there's that sweet spot, this window, this therapeutic range.
And just let me remind myself, I suppose.
Please.
I think in the Graham Rook, who developed Mycobacterium vaccae for TB to boost the immune system for TB protection, all the studies were being done in Africa. In order to boost its efficacy, you could see there was some. They increased the dose, no difference, but he did what enters into this territory. He started reducing the dose, and a tenth of the dose, the response was far, far better. So it's another low dose. So the vaccae and obiense are a fraction of the dose in which they were initially developed.
Really?
So once again, it is, and so these things are what I call bell-shaped curve.
Yeah.
So you get to the top, and that's low dose naltrexone is at the top. Give more, you lose the benefit, just like that. So it's a biological, actually. If you got to classify it, it's a biological, because it's not the more the merrier.
I mean, many people watching will realise what a massive pandemic tuberculosis is. I mean, it's huge in Africa and India and Asia, and causes indescribable suffering. I mean, I saw a child with tuberculosis meningitis in India, and you could just see this child was suffering this most intolerable pain and agitation from the meninges. And even if being treated, it would take, with traditional treatments, it would take weeks for the inflammation to go down. If we could protect people from against this, it's hard to describe how massive an opportunity this is. Well, it's simply being ignored by international agencies, as far as I can see.
Yes, and we'd like to know why, when they suck so much money out of our governments for vaccinating the world, Gavi, CEPI, United Nations, they're all got Bill Gates behind it, and Fauci working away there, when there is something really simple. And I know they've all seen this data, because emissaries have given it to these people, and they just basically show no interest at all. The M. vaccae, what needs to be appreciated about it, that they did a study, and I think it was led by people from University College. They did it in Africa, in HIV, the height of it. They gave the vaccae to people HIV positive, there was no treatment, in tuberculosis areas where there was a high risk of tuberculosis.
And of course, the HIV and the TB together are real bad double whammy,
real
aren't they?
Unbelievable. But they had a highly significant reduction in TB infection in those given